Alright guys, so welcome to Subu Medicine, and today we'll be talking about the pathology, alright? So, in this lecture, we're gonna briefly talk about what pathology is, and also we're going to clear some of the concept when it comes about the definitions of it, alright? So that's what we're gonna do in this lecture. So, we're discussing about pathology, but we need to know what pathology means, right? So let's write down what pathology is. So you have a pathology, right? And remember guys, this pathos is referring as suffering, all right? And then you have a logos, all right? That means study. So basically, in a simple term, pathology means study of suffering, all right? And this pathos, which means suffering, it's a Greek word, okay? The Greek word of pathos means suffering, all right? So now we're studying of suffering, all right? So whenever we study about this pathology, we always have to look at each disease, its conditions in 4 different fragments, all right? 4 different sections, all right? And let me write that down. Look, whenever any disease, all right, first we need to know what is the, let's say, etiology of this, all right? And I'll define what this means. Second, you have to know something called pathogenesis, all right? And third, you have to look at the morphological changes, all right? And we'll define what that means. And fourth is the clinical expressions, all right? So every disease, we have to talk about what is the etiology of it, what is the pathogenesis of it, what are the morphological changes, all right? And then we have to talk about the clinical expressions, right? Look, and whenever we talk about the etiology, right, what is etiology? Etiology basically means what is the underlying cause or modifying factor that initiated a disease, or let's say a tissue damage, which is going to result into the, let's say, a disease later, all right? So basically, whenever you think of etiology, think about what cause, what are the modifying factor that is initiating that disease. That's what basic etiology basically means, right? And once you know, let's write down your cause. Okay, let's say you have a cause, right? Let's say this is A is a cell, right? Because remember, right now we're talking about us, right? Human beings. So we are made out of what? We're made out of cells, right? So that means that if anything there's going to be problems happening in cells, then there is going to be a problem, right? So basically, we're talking about our tissues, our cells, our organs, all right? That's what we're talking about here. So let's say this is one cell right here. If some kind of like stress in the cells, these cells are gonna go certain changes. Maybe let's say it become this cell. Then after that, maybe it will become this cell, right? So look at, what is the mechanisms of this disease? What is the development process of the disease? What is the sequence of event that's happening, okay, from this A cell to get into this B square cells, from the B square to, let's make this as a, let's say this type of cell. What are the sequence of events that are happening? What is the mechanism that is taking place to get into this disease? That is what basically pathogenesis basically means, all right? Because once this happens, the sequence of event that takes place and cells modifies, all right, the cell, let's say, progressed in a different layers of the cells, right? It changes its size, it changes its shape, right? It's going to happen there, right? And then when you change the shape and size, or become larger or smaller, there will be some sort of reduce of the functions of the cell too, right? So all this event that is taking place, right, to lead into the disease, that is what I call pathogenesis is, right? So once you have this disease right here, okay, you see what is going to happen is there is going to be morphological changes, as I said, shape and size, okay? Whether it's going to be firm, it's going to be hard, it's going to be soft, all right? That is also, those are the morphological changes that you get to see, all right? And in here, we also have to talk about the morphological changes. We also have to talk about, the color also. Like what is the, if we let's say stain with a certain color, does it change? What kind of shape you get? What kind of color you get? That also plays a very, very important role to determine the exact what is the disease. All right. So and then after that, let's say once you have this morphological changes, then you also get this something called the clinical expression. What does this mean? Clinical expression basically means is that this disease of cause, what is the sequence of event that took place, all right? And then after that, what is the shape? What is the color? What is the form? Is it hard? That is all comes to morphological changes, all right? Then after that, what kind of clinical symptoms the patient or individual is having, all right? So that we have to look at all of that in order to, whenever we're looking for any pathological disease, all right? And whenever we talk about clinical expressions, I'm not saying physical signs and symptoms. Sometimes there are a lot of times, you could have a virus, but then you may not have a physical signs and symptoms, but you may have a clinical symptoms or clinical expressions, meaning that you can find this clinical expression via lab reports, through blood, through urine, right? Or X-ray. So that's what I'm talking about, all right? I'm not necessarily meaning the clinical expression. I'm not necessarily saying that it has to be physical signs and symptoms, all right? It will lead into physical signs and symptoms if the disease progresses, but there are conditions or diseases there that you may not have a signs and symptoms, physical, all right? But then you are, let's say, if you do a blood test, you see their, let's say, antibodies or their antigens present in your body, right? So that's something that, and antibodies obviously, that you get to see in your blood, right? And that's called clinical expressions, okay? So, this morphological changes, let me put a little stress on this, all right? Whenever you look at the morphological changes, right? First of all, the first thing first, right? First you need to know the anatomy of it, right? What is the anatomy? And the morphological changes usually, we divide in two parts. We call them a gross part, okay? We say gross anatomy, all right? And then gross structures, we'll say. And the other one is more like a, microscopic. And we have to talk about this. Whenever we look at the gross, we look at the size, all right? We have to look at the shape also, all right? We have to look at the, is it solid? Is it form? It is, compare, and you have to compare to normal, right? You have to compare to normal. Is it swollen? All right? Does it have a fluid inside? All right? So you have to look at all of that. And after that, all right, in here we'll just say solid, or we'll say form, all right, we'll say soft, all right? All of that also plays a role in it. We also have to look at the temperatures, all right? All of that. And then after that, we also have to look at one more thing. Size, shape, solid, and then you have to look at the color, right? The color. Because remember, sometimes, and this is physically you can see, because sometimes what happens, if there's a swollen, if it's more reddish, and you always have to what? Compare with the normal. So that's what, it's really important to know the anatomy of it, right? So, this is you looking for more grossly, all right? And, look, whenever you look at the size and shape, all right, and then you look at the solid and form of color, you also, first thing you have to do is what? You have to identify the organ too, right? Which is this? Which organ is that you're looking for? That's the first and first, right? And after that, you look at this and you compare with the normal. Then after that, okay, in order to be let's say, correctly identify, okay, what is the disease or what is the problem, we have to look at the microscopic. We have to look at microscopically, meaning that we have to use certain stain or let's say certain immunohistochemical antibodies in order to identify which cell or what part of the cells is problematic so we can treat it, all right? So there are a couple of the most common, which are gold standards, I'll just write down here. What's this called? Hematoxylin, all right? And eosin. Or simply you can say H&E, all right? This is very common, that's used all the time. Because the reason why is because hematotoxins, this is very, very important. These guys are very, very important for binding to calcium salts or even nucleic acid, all right? So that's also why the hematotoxin becomes very, very important. right? Remember, we have a lot of this DNA in our body, right? And then eosins is basically, it binds to like thyroid, the colloid, or any sort of that has like amino acids features, like for example, proteinaceous materials, or you could even say red blood cells, all right? So these guys, eosins binds into that. And whenever it binds to, let's say, red blood cells, usually, look, it can give you pink or reddish color. And this hematotoxin will usually give what? Is it purple or bluish color, all right? It's very, very important. And it's really important to distinguish between that, hematotoxin and eosins. The other ones, like for example, like PAS, which is called periodic, let me write down here, periodic acidic SIF or SCIF, whatever you like to call it. This is called PAS. And this PAS are very, very important for binding to the carbohydrates content materials. Like for example, if you're looking for, let's say I'll give you the examples of, let's say, like a tinea pedis. That is in fungi infections, right? Or in a simpler term, they also call this as an athlete's foot, right? So there, when the fungi cell membranes has a lot of this carbohydrates materials there. If you want to stain that, okay, you want to use the PAS, all right? So whenever there's a lot of this carbohydrates content materials out there, you probably want to use this, all right, the stain. Other one that's right down here, okay, we can see Congo red. This is usually used as stain for if there is a suspect of the amyloid deposit, maybe in your heart, all right? So you use Congo red. Okay, let me write another one. Something called the Persian blue, okay? This Persian blue, P-R-U-S-S-U-I-A-N, Persian blue, this stain is very important for binding to iron, all right? It's very important, this Persian blue. All right, I'll give you and also you have to remember the gram stain, gram stain for the bacteria and the gram-positive bacteria, all right? Those ones are very important too. And also, you don't want to forget the immunohistochemical. Like, for instance, they often use something called a cytokeratin to look for the epithelial cells. Or like a dasmin, all right? The dasmins are for what? The scata muscles, smooth muscles, like cardiac muscles, they use that. And obviously, you also have to do those, the TEM, like trans-electron microscope. These are some of the stuff I'm just mentioned because as we talk, all right, we'll go more detail about this too. And obviously, which I don't want to forget, for even those molecular changes, we look for what? Like, for example, if you have AIDS, we do those, southern blotting or western blotting, right? The DNA. And then also the PCR. So that's also important. So these are some of them, all right? As I said, this is just an overview, all right? We'll go more detail about as we talk about the diseases, all right? But these are general concept, right? Because this is you have to look at, whenever we look at the morphologically, we have to look at the grossly and also microscopically in order to see, all these different changes, all right? Or correctly identify or suspect what the disease is, all right? And you use all the different skin colors, all right? Now, so again, pathology, every time we talk etiology, we're going to talk about etiology, pathogenesis, morphological changes, and also whether the disease is able to give you any clinical signs and symptoms, all right? So that's something we're going to talk about all the time. Now, here I drew as a one cell right here, right? And then, this is a one cell. Let's say this is a normal cell, all right? Meaning that this is a normal. Whenever we talk about normal cells, that means this normal cell is functioning at its steady state, meaning that it is maintaining its internal constant environment, or it is maintaining constant environment. What does that mean? What does that, it looks like I'm saying the definition of something, right? Yes, I am. That is called homeostasis. Every cell needs to maintain something called this, steady state or a certain threshold or homeostasis. Steady state or homeostasis, which basically means you're maintaining the constant internal environment, all right? Because if somehow, let's say, this cells gets certain stress, right? Let's write down stress. And the stress could be anything. Let me just share. It could be, let's say, hypoxia, low level of oxygen. all right, hypoxia, low level oxygen. It could be because of anemia, that'll come in hypoxia, or it could be like physical agents, right? Like for example, like, just like, you hammer the hand or something, all right? It could be like infections, like any fungi, all that kind of stuff. It could be because of nutritional imbalance, all right? Because your cells needs, your cell needs what? Nutrition, right? In order to work, right? Anyways, so any of the stress, right? It could be oxygen deprivation, it could be physical agents, it could be infection, it could be nutritional imbalance, all right? It could be immunological reactions, all right? It could be chemical agents, all right? Anything, like for example, you spill over the acid, that would be chemical, right? So anything that, causes stress on the cells, all right? Anything, when that causes the stress in the cells, it depends on, whenever the stress is also, look, the cause of stress, it also depends. What is the duration? What is the severity of that cause? All right? Because what happens is that the cells, whatever the stress is there, right? The cells will sort of like try to adapt it, all right? But the cell can adapt it in a way, how much it has energy, or what is its capability, whether that cell can able to handle that stress or not. So it depends on what kind of stress is that. The stress is intense and severe that, the stress cannot handle it. Then it will directly go to this, something called, cell injury. It will call cell injury. And this is called cell injury, right? Because it's direct trauma or head, let's say. And this is called cell injury, right? Or let's say, the duration is not much, or the severity also, the intensity of that stress is very, very low, or let's say hormonal imbalance is also very low. That means what is going to happen is that, let's say, the cell will go something called because cell can handle it, cell can adapt it, right? Adapt that changes. Now as soon as when we talk adapt, what is adaptation? That's what we're talking about. Adaptation basically referring as the cell can adapt, okay, whatever the stress or whatever the cause is there, and if you remove that stress or stimulus, whatever it is, whether the cell can able to, change its size and numbers and functions, okay, and come back to the, let's say, normal level like this. All right, and if the cell can able to come back to the normal level after you remove the stimulus, then we call them reversible. This is called reversible. Whenever you think about reversible, think about life. The cells is able to work now, right? And then whenever, let's say the cells is not able to adapt at all because maybe the stress is continuous or it's a progress for a longer period of time. So when there's inability for the cell to stress, let's say inability for the cell to adapt, this cell is going to go, this is called cell injury. All right? Injury. Now, even the cell injury, when the cell is injured, even injured cells, okay, it can still go back and be reversible, by the way, all right? For example, I'll give you examples of liver. Let's say if you remove a small piece of liver, a liver has what? Liver can regenerate, right? So it can go back to the normal state, or it can meet its steady state, right? And that is also called a reversible change. Again, reversible, always think of life. You're going back to the normal life processes, all right? But sometimes what happens is that this cell is like traumatized in such a way, there's no way that cell can go back to normal. Cell can no back and meet its, cell cannot go back and meet a normal state or its steady state. In that case, cell has only like couple of ways the cell can do. The main two ones are, let me write down here. Either cell can go this process, which is called irreversible, meaning that whenever you think of irreversible, okay, irreversible, think about death. All right? If there's a severity, durations are longer, and the cells cannot go back to this normal state, then cell can do irreversible or death. It will go to the path of death, all right? And cells will choose either it will go to this process, which is called apoptosis. What is apoptosis, guys? it's well organized cell death. We also call it programmed cell death. All right, this is, and then although it can go to this process, which is called necrosis. Both of them are cell death, guys. Right? Both of them are cell death. Both of them are irreversible. They both are leading into like death of the cell. But the one way is apoptosis, meaning more organized, more well-mannered. Okay, they are more fragmentatedly, they're programmedly, they're organizedly, they die. Okay, they don't spill over anywhere. Other one is that necrosis, the cells dies, but then when the cells dies, it spills over to your vascular compartment, and usually what that lead into causing inflammations. There is one biggest difference between apoptosis and necrosis. There are going to be other differences too, but one of the biggest differences is that necrosis causes inflammations, the apoptosis do not. That's one of the big differences, all right? So that could happen. Other thing that could happen is called subcellular alterations could happen, which basically means is that there is a change in the, which means that there is a change in the, there's a structure of protein that is, the cytoskeleton, all those actin, myosin, all those microtubules, they are what maintain the structural shape of the cell, right? Sometimes, cell can be irreversible and you see the subcellular alterations, which basically means there's a change in the structural proteins, all right? There's something that I'm giving you extra here. But remember, again, if there's a stress here, okay, cell can adapt, all right, adapt by increasing the size, increasing the shape, increasing the number of those cells. That's how it can adapt, right? And then if the cell is injured for a very, very longer period of time, or the cell doesn't have the capacity to handle it, or capacity to adapt it, or cell does not have any capacity to regrow or increase the number of size cells or multiply, then the cell will go something irreversible reaction, meaning that it is choosing the death pathway. So in that way, the cell can do two ways. Either it can do the main two ways, either apoptosis process or it can do a necrosis process. All right? Now, and then as we talk about diseases, you'll know more about this too, all right?